Okay, so today I'm going to talk about vision. Vision is, in my opinion, the most impressive sense that we have, and I think those of us who see take it for granted, but we're interpreting light bouncing off of distant objects into a mental representation of the world in which we live in. We all have a virtual reality in our head, and that's pretty fancy for a bag of bones and blood. Not only can we do this, but we actually have vision that works in a huge variety of light levels. So the difference in the number of photons between the lowest level of light that we can see in and the highest level of light we can see in is over 9 orders of magnitude. And this is what I'm interested in. How does vision work in such a large range of light levels? More specifically, how does the retina encode one of the most basic visual features it detects, contrast, at those light levels? But I'll get into that more in a little bit. I first want to just briefly, ever so briefly, talk about the retina so that we're on the same page. So the retina, of course, is part of your central nervous system. It is a neural circuit comprised of 5 classes of neurons shown here in different colors. They're all networked together in such a way as to begin processing light information before it's even sent to the brain. So on the input side of this circuit, we have photoreceptors, which directly interact with light and signal this interaction through the bipolar cells here in green to the output side of the retinal ganglion cells in blue. And this vertical flow of information is modulated in a lateral way by horizontal cells and amacrine cells, adding complexity, but I don't really want to talk about those. Rather, I'd like to focus on the output side, those retinal ganglion cells that directly connect to your brain, because it is their voices, the action potentials that are sent down your optic nerves, that are the sole source of visual information that your brain gets. And this is further complicated when you consider the vast diversity of cell types within each neuronal cell class in the retina. So it's not just a direct flow of information. Rather, visual information diverges and converges into different synaptic patterns, all funneling down onto not one, but rather over 20 different types of retinal ganglion cells, each depicting some aspect or aspects of your visual world. And what each retinal ganglion cell type depicts, what feature it picks out, is not fully known, but we do know that all retinal ganglion cells at some level encode contrast. And contrast is very simple. It's just a change in brightness, whether it's an increase or a dimming of light. And so all of those different types of retinal ganglion cells can actually be categorized into 3 main groups: on, off, and on-off retinal ganglion cells, so named for their preference for either responding to the onset of light, brightening or positive contrast steps, the offset of light, dimming or negative contrast steps, or responding to both brightening and dimming. And so I have 3 example responses from 3 different cells, one from each type here, shown in 2 different ways. First, a spike raster, and then a contrast tuning curve. And so the spike raster is actually pretty simple. There are 2 different types of stimuli, an on stimuli and an off, so brightening and dimming. And then this is on the x-axis, time away from that particular stimulus. And the y-axis is just a repeat recording from the same cell. And each dash represents a spike or a signal that this cell would be sending to the brain. And so this on retinal ganglion cell reliably and robustly responds to increases in light, but not decreases in light. The off has the opposite behavior, and on-off responds to both increases and decreases. Now, contrast can change not just in polarity with positive and negative contrast steps, but it can also change in magnitude. There can be small shifts in contrast or huge shifts from complete blackness to blinding white. And so we can plot a cell's response to all the different types of contrast, magnitude and polarity, to get a sense of its tuning curve for contrast. And so for these particular cells, I have those plotted below. And so this, again, is an on cell in that it only responds to positive contrast steps. And this is an off, only negative, and then both negative and positive. Now it spans from -50 to 50 because there is a 100% range on the screens that we use to simulate retina. And we start in the middle, and then we can go darker or brighter. So it's either positive 50% or negative 50, if you were wondering. Okay, so we're on the same ground about the retina. We've talked about contrast, so we can go back to my question about how contrast is encoded by different retinal ganglion cells at different light levels. And so to do this, I use a tool called a multi-electrode array, shown here with dramatic lighting, on which you can place a dissected retina and record the extracellular voltage changes that those retinal ganglion cells produce in response to different light stimuli. And so here's the readout where you have different rectangles corresponding to different electrodes and the activity upon them. And so with this recording technique, I have a high throughput way of recording from many different types of retinal ganglion cells all at the same time. I can, on a good recording, record from hundreds of different retinal ganglion cells in one day. I show many different stimulus patterns to characterize the responses, and I show them at 5 different light levels, shown here, from 1 to 10,000 rod isomerizations per rod per second, which I know means very little to you. So look at the cartoon, the analogous natural light levels. The lowest light level I show is about the same as a crescent moon on a nighttime sky, and the brightest light level is a little brighter than in here, a slightly overcast day. All right, so I showed many different types of stimulus, and you can learn many different things. I don't have time to talk about all of them. The first stimulus that I will talk about, though, is just a basic contrast step, which you start with some mean middle background level of light, I sub b, and then you increase or decrease, and we've seen the results of this before with the raster plots and those contrast curves. So I found over all the different light levels, starting with the lowest to the brightest, that most retinal ganglion cells consistently and faithfully encode their preferred contrast, here showing an on-ganglion cell. And not much changes. We've known for a while that retinal ganglion cells become faster at responding to their preferred stimulus. As you can see here, it's over to the left a little bit more. But beyond that, nothing too surprising, except for a fifth of the retinal ganglion cells of which I recorded, that although they are reliable, they are not faithful. Depending on the light level of the stimulus that I am showing, they encode different types of contrast, and this was novel. So I looked at this a little further, and I was able to actually categorize them into 3 distinct functional groups, different types of switching retinal ganglion cells, and I'm just going to walk you through those 3 and their responses to contrast steps. So type 1 starts with an on preference and goes to a slight off preference. So you can see here at the lowest light level, remember a nighttime sky with the crescent moon. It really only responds to increases in brightness. But then as you get brighter and brighter, it responds more and more to decreases in brightness as well. And so this is one example cell. This is also from that same example cell, the same contrast tuning curve. And this is the population data, which includes error bars, though you cannot see them, because from cell to cell, tissue to tissue, all of the cells in this group are pretty similar. That holds true for the second type as well, which looks similar at first glance. It is also on preference to off preference, starting here and going down to there. But there are 2 major distinctions, major significant distinctions, that separate it from type 1. The first is that it starts responding much more robustly to the opposing contrast stimulus than type 1. And the other distinction we'll get to in a moment, but requires a different type of stimulus. All right, before we get to that, though, the last type of switching retinal ganglion cell that I found was the opposing behavior off preference to on preference. So again, you can see at low light levels, it has a pretty robust response to decreases in light, but that goes away completely when you get to the brightest light level that I showed. And again, for this particular cell and for all of the cells in this group that I found. The second type of stimulus that I'm going to talk about today is called a drifting grating, which is what it sounds like. It is a grating that you drift across the retina. You can do this in any direction you like. I did it in 8 different directions. And this is because we know that some retinal ganglion cells are direction selective, in that they respond very robustly to stimulus moving in one direction, but they will not respond to stimulus moving in the other direction. They have a true null. And so, as you might be guessing, we found that one type of our cells was actually direction selective, type 2, further distinguishing it from type 1. So I'm showing you this data in 2 different ways again. This is the average of all the different cells with normalized spike rate on the y-axis and direction of stimulus on the x. And we have centered it such that 0, the origin, is the direction that elicits the maximum firing rate from these cells. So it will always have the highest firing rate. And you can see that type 2 is the only with a true null direction. And we look at this again with heat maps, where red is the max firing rate, white is no firing. And again, we're centered such that the middle is the direction that elicits the maximal firing rate. And we can see that this is the only one that has opposing directions of no activity. The others fire at all directions. So this was cool. So I set out to try and characterize how different types of retinal ganglion cells encode contrast at different light levels. And I discovered that some of the retinal ganglion cells, in mouse eyes at least, do not do so faithfully, and that they actually switch the type of contrast that they encode based on the light level. And so from here, there are many different ways that we can go. One obvious one is to try and correspond those functional types that I've discovered with morphological types that are already known. MEAs are great for functional data, but show almost no morphological data. So this line of experimentation would require single-cell patching and filling cells with dyes to see what they look like. We also are trying to push what the MEA is capable of doing. We can record spiking events, but we can also record population voltage changes from the entire retina, which corresponds to a very old ophthalmology technique called an electroretinogram, which gives you more information about all the other cell types in the retina. And we're going to use this, and this is just a teaser, to try and screen a bunch of molecular candidates involved in synaptic circuitry in the retina. But I can't talk about that today. So that is it. Thank you. I'll take any questions. Yeah.
So with your different types, I was wondering if they have, if they actually interact with other, like rods or cones specifically, if they have different preferences for other cell types that they interact with.
So I can't say definitively which ones they interact with and what ratios. They have to interact with both rods and cones because the range of light that I use spans rod-dominated and cone-dominated pathways. But the rod pathway and cone pathway are connected to all retinal ganglion cell types. That answers your question. Uh-huh.
Is there a refractory period, and is that different for these ganglion cells?
For which they will not fire again, spiking rates? Yes. As if they're different for the different types of retinal ganglion cells, I'm not sure. I'd have to go and look at that. Yeah.
Does the proportion of each of the types you've discovered change with age from P0 as to when the visual system fully develops to open eyes?
So we only look at adult. So I can't say. Anyone else? Oh, yeah, sorry.
Does the type of shifting vary in the culture slices depending on the other types of retinal ganglion cells that surround it? So is like type 2 surrounded by other type 2s or type 1s?
So all different retinal ganglion cells in the retina have a mosaic patterning, so there's even spread across the retina, more so in mice than us, actually. So there should be an even spread, but I always use the same area of the retina, so it should be pretty consistent from recording to recording. Okay, thank you.